Okay, in this recording, I'm going to give you a bit of an overview of proteins and explain a few things about proteins, what they're used for, what their structure is, and how we talk about them in the context of VCE biology. So to start us off, we call a protein a polymer or a biomacromolecule. So proteins are biomacromolecules, and most biomacromolecules, so of which we have proteins, nucleic acids, lipids, and carbohydrates, all of these biomacromolecules, except for lipids, are examples of polymers. So a protein is a polymer, which means it's a large molecule composed of many smaller molecules called monomers, so single units. In the case of a protein, the monomers are called amino acids. And essentially, a protein is a whole heap of amino acids all joined together to make a big long chain, which we'd call a polypeptide chain. And then that chain goes through a process of folding to become a functional protein. When we talk about proteins, there's heaps and heaps and heaps of different types of proteins, and you couldn't possibly remember all of them. But what you guys do need to be able to do is you need to be able to remember a good few examples of different types of proteins and some different functions that those proteins carry out. So in terms of proteins, if you have a look at this table here, which gives us a pretty good snapshot, we've got some proteins involved in movement, some proteins involved in giving the cell its structure. Some proteins have a role in catalyzing chemical reactions. We call those proteins enzymes. Some proteins are hormones, which have a role in cell signaling. Other proteins act on the surface of a cell as receptors. Neurotransmitters are a type of protein, once again involved in cell signaling and communication between cells. Immunoglobulins, which you guys probably know as antibodies, are a type of protein, and they help your body to fight off pathogens and infections. And then there's also some other types of proteins that act as poisons and toxins as well that can sometimes enter your body. As I said in the little intro, a protein is a polymer, which essentially means it's a big long chain of amino acids. So a simple way to represent a protein down the bottom here, a circle could represent the protein, a line represents a chemical bond, the circle represents the amino acid, the line represents a chemical bond, the next circle would be another amino acid. So in its simplest form, which we call its primary structure, a protein is a big long chain of amino acids all joined together. Now, this is a pretty convenient way to draw it, but you guys need to know a little bit more about the structure of an amino acid than just saying it's a circle. And if you got asked to draw a diagram of an amino acid and you just drew a circle, that's exactly how many marks you would get for your answer, 0. So what each of these circles is representing is a molecule called an amino acid. And that is this picture up the top here. And this whole molecule here makes up what's called an amino acid. I would expect you guys to be able to draw this diagram, definitely. And I'd expect you to be able to recall how many of each type of element or atom is in this molecule as well. So to make that a little bit more simple for you, our amino acid is in, we can break it up into different parts of it. So in the center, we have a carbon. Coming off that up the top is a hydrogen. Coming off that down the bottom is what we call the R group. We'll talk about that in a minute. On the left here, we have the amino group, which we call H2N, because there's 2 H's, 2 hydrogens, and 1 nitrogen. On the right over here, we have the carboxyl group, which we refer to as COOH, 1 carbon, 2 oxygens, and 1 hydrogen. The good thing about remembering the structure of an amino acid is that, yes, we have 20 different types of amino acids, but each of those 20 different types have the exact same structure. The only thing that differs is the R group, which is representing another molecule which attaches here where the R is representing. And that's the single only thing that makes one amino acid different to another. So we have the same general structure, but each individual amino acid varies in the type of molecule that's attached here at the R group. So as I said, essentially to make a protein, we take this amino acid, we join it to another one, and another one, and another one, and another one. And when we're doing that, the amino group of one amino acid is always joining to the carboxyl group of the other amino acid. So if this is our carboxyl group over here, the next amino acid's amino group would join on to there. It's A for amino, by the way. This here is a pretty busy and probably intimidating slide for you to look at. Take comfort in the fact that you do not need to remember this slide in any capacity. You certainly don't need to draw all of the different amino acids on here, but what it's trying to do is it's trying to show you that these are our different amino acids. So often we abbreviate amino acids to a 3-letter word, so gly for glycine, ALA for alanine, VAL for valine, etc., etc. When we do a bit of work with the genetic code, you'll see that that's what the genetic code has in it as well, those 3-letter symbols. What you can take away from this diagram though, is that if you look at every amino acid, it does represent that general structure. The carbon in the middle, the hydrogen up above it. So I'm looking at this one here. Carbon in the middle, hydrogen above it, carboxyl group to the right, amino group to the left, and then down the bottom here is where the R group is. Glycine has a hydrogen there. If you compare that to alanine, same general structure, but instead of the hydrogen being attached as the R group, it has 3 hydrogens and a carbon, so CH3, and that difference in the molecule that's attached makes it a different amino acid. That's what gives amino acids different properties. So you can see in this row up here, all of these amino acids are non-polar. So all of the R groups are non-polar R groups, whereas this row here, all of the R groups are polar R groups, so they make the amino acids polar. Whereas down here, all of the R groups are electrically charged, so they make those amino acids have an electrical charge. So now we can appreciate the structure of amino acid and the idea that they join together to form a chain. What we also need to know is, well, how do they actually join with one another? So what's that process? Obviously, a chemical reaction takes place that results in a bond between one amino acid and another amino acid. And just so you know, we call that a peptide bond. So if I go back to my simple diagram, I'll draw it up the top this time, with my circle representing my amino acid, another circle representing the one it's joined to. This bond here is what we call a, I'm not going to be able to write it with this little pen, I'll do a PEP, P-E-P. It's called a peptide bond. So to form a peptide bond, a chemical reaction needs to take place. We need to know what that specific chemical reaction is, and it's called a condensation reaction. So amino acids are joined together through condensation reactions. If we break that word down, condensation, the other context we think about condensation with is water droplets, and that's exactly why it's getting its name. So when we join one amino acid to another, what's actually produced and released is a molecule of water. Hence we call it a condensation reaction. So adding to my simple diagram here, when I join one amino acid to another, to form that peptide bond, we actually form and release one molecule of H2O. Therefore, condensation reaction. Fun fact for you, every biomacromolecule, nucleic acids, lipids, carbohydrates, and obviously proteins, all of their monomers join together through condensation reactions. The diagram here on the right and the one on the left are showing the same thing, just in different ways. We might follow the one here on the right. So we've got an amino acid here. It's called amino acid 1. You can see its general structure. Amino acid 2, obviously the same general structure. Like I said before, if we're joining two amino acids together, we always join the carboxyl group of one amino acid to the amino group of the other. So we've got our carboxyl, COOH, and our amino, 2 H's and an N, H2N. And what this is showing you is when those two amino acids come together, what actually happens is a hydrogen and one oxygen from the carboxyl group of one amino acid pairs up with one hydrogen from the amino group of the amino acid it's attaching to, and that gets released. And if we take 2 hydrogens and 1 oxygen, obviously we've got H2O. The bottom picture here is showing the result of that. So we've got our amino acid here, amino acid 1. It's now missing a hydrogen and an oxygen. Joined to amino acid 2. It's now missing one of those hydrogens. But we've got ourselves a peptide bond, and we've released a molecule of water. So we would call this a dipeptide because it's 2 amino acids joined together. A polypeptide would be more than 2 amino acids joined together. So condensation reaction. Question down the bottom here, what type of reaction do you think would break down a protein during digestion? What I'm getting at there is the fact that if to join 2 amino acids together we release a water molecule, what would happen if we add a water molecule into an amino acid, to a dipeptide? We would get a hydrolysis reaction, hydrolysis, lysis meaning to split, hydro meaning water, and we would break it down. So obviously we ingest proteins when we eat food. We want to break those down into individual amino acids so we can absorb them back into our bloodstream. To do that, we use hydrolysis reactions to break them back down. So let's continue building up on what we know about proteins. So far we've got this idea of amino acids joining together to form a chain. But a chain itself, so this little diagram I keep drawing, that by itself is not a functioning protein. To get that chain of amino acids into a functioning protein, we need to fold it up into a 3-dimensional shape. And at that stage, then we've got ourselves a functioning protein. So the next thing I'm going to take you through, protein folding. Is our process where we go from our chain of amino acids to our functioning protein. And protein folding happens to all proteins, and each protein goes through at least the 3 stages of protein folding, and then for some proteins there's also a 4th stage of folding. So we've got our first stage, which is called our primary structure of a protein, and then we move into our secondary structure of the protein, which is our 2nd stage. 3rd stage of the folding process is the tertiary structure, and at this point some proteins are fully functioning and off they go and do their role, but for some proteins they also enter a 4th stage of folding called the quaternary stage. So what we'll do now is we'll go through each stage, have a brief look at what's going on, but we'll also unpack this in class and do some modelling on it as well. Let's start up the top here. So stage number 1, primary structure of a protein. Quite simply, that is a polypeptide chain. So the primary structure of the protein is a protein which is just a chain of amino acids joined together by peptide bonds. The important thing to think about with primary structure is that the specific sequence the amino acids are placed in determines the type of protein it will become, because it determines the way the protein will fold and therefore function. So the sequence of amino acids is really important. As you guys would know, we have 20 different amino acids, and we arrange those 20 different amino acids in all sorts of different ways to get all of our different proteins. If we had 2 proteins, for example, let's say each protein was made of, let's say it's really small, like 10 amino acids long, and the second protein was made of the same 10 amino acids, exactly the same 10 amino acids, but in the first chain we had the amino acids in the sequence like amino acid 1, then 2, then 3, then 4, then 5, 6, 7, 8, 9, 10. In the second protein we had them in a different sequence, so maybe 1, 3, 5, 2, 4, 8, 7, something random like that. Though they're the same amino acids, because they're placed in a different order, therefore the protein will fold up differently and become a different protein. Let's move into the second stage of folding, which is the secondary stage. So what happens is the amino acids start to interact with one another, and hydrogen bonds form between some amino acids, and by those hydrogen bonds forming, the chain starts to fold up a little bit, and it will fold into a couple of different but specific structures. So our first one we call the alpha helix. So the chain folds into what can be a helical type structure, or the chain might fold into what we call a beta pleated sheet, where it folds into this sort of pleated type structure. They're definitely our 2 main ones. You can also get them fold into what's called like a random coil. So it's not an alpha helix or a beta pleated sheet. It's sort of like a random shape. But the ones we talk about in VCE are alpha helix or beta pleated sheet. So if a question said describe the secondary structure of a protein, you would say the protein would be in the form of an alpha helix or a beta pleated sheet. If the question said what causes those structures to form, you would say hydrogen bonds between amino acids. And just so you know, it's not the case that one protein is completely made up of alpha helixes or beta pleated sheets. You see different regions of a protein being in the form of alpha helix and beta pleated sheet throughout pretty much every protein. After our secondary structure, the protein folds up even more into its tertiary structure. And what happens at the tertiary level is the amino acids are still interacting with one another. And what's occurring here is specifically the R groups of particular amino acids start to form bonds with one another. So some R groups will bond with one another through an ionic bond, so opposing charges. Some R groups will bond with each other through a disulfide bond, which is through a sulfur group between some specific R groups. Some of them will bond together through hydrogen bonds as well. Either way, no matter which way they're bonding, what it's causing to happen is it's causing the protein to fold up even more, and it forms a 3D structure. So if you got asked to describe the tertiary structure, you'd say the protein is in a specific 3-dimensional configuration. Why is it in that configuration? Because of bonding between specific R groups. At this stage, at tertiary, it is a 3D protein, so it is a functional protein for most, or for many. And some proteins, once they get to the tertiary level, they've finished folding up, they'll go off and they'll do their job. But for some proteins, they enter a 4th stage called the quaternary stage, or the quaternary structure. And what's going on in the quaternary stage, there's no more folding as such, but what quaternary is it's when 2 or more tertiary proteins join together to form a larger protein. So for example, hemoglobin is a protein on our red blood cells. If you look at this picture of hemoglobin, it's got one 3D protein there, a second one there, a third one there, a fourth one there. So hemoglobin is actually a protein composed of 4 polypeptide chains that are all joined together. So we say quaternary is any protein composed of 2 or more subunits, which means 2 or more polypeptide chains. Antibodies are another good example. Of all of our proteins that we have in our body, though there's so many different types, we can categorize them into one of 2 groups. So proteins can be either fibrous or they can be globular proteins. And it's pretty common to get some sort of question that gets you to compare between the 2 of them. And the things you would be comparing is the shape, their properties, and what their function actually is. So a fibrous protein is a protein that sort of has a shape where it's got lots of parallel polypeptide chains, and they form what looks like fibers or sheets. So they're really strong. So their role is in structure. Good examples are collagen, which is drawn down the bottom here. Other examples could be keratin or elastin, other proteins that have a role in giving in the body's structure. So as you can see, when I say they're parallel polypeptide chains, you can see there's 1, 2, 3 polypeptide chains running sort of parallel with one another, intertwining, forming what even looks like a pretty strong sort of structure. Properties is that fibrous proteins are not soluble in water, which is a pretty good thing to have when it's got a role with structure. We wouldn't be very good if our structural proteins kept dissolving in our bodies, which is predominantly water. This is compared to a globular protein. So globular protein, when I think glob, I think blob. So it sort of looks like that blobby sort of globular shape. So it's not the nice, neat, parallel polypeptide chains. So it forms pretty much a sphere. And that's what we're seeing down here with hemoglobin, sort of a sphere type shape. Globular proteins are soluble in water, and the tertiary structure, their 3D structure, is really critical to what their function actually is. With globular proteins, their function is typically like a catalytic or a regulatory role, which means they have some sort of role in chemical reactions. So enzymes are a really good example. Quickly talking about another category of protein, which would be a conjugated protein. Now, it still would be like a conjugated fibrous protein or a conjugated globular protein. So what it means is some proteins, they're just made up of the amino acids folded into their shape, and that's it. That's the protein. But we also have some proteins in the body where their functioning structure is not completely just amino acids. They've actually bonded with some other type of molecule that's not an amino acid as well. So we say conjugated proteins are ones that contain amino acids that have conjugated, which just means sort of joined up with other groups. So a nucleosome is a term we use for your DNA in your cells when it's wrapped around these proteins called histones. And histones, just so you know, play a role in helping that DNA condense into a chromosome. Hemoglobin over here, as you guys would know, it's got 1, 2, 3, 4 polypeptide chains, but that also contains this little molecule here, which is called a heme group, which is an iron-containing compound. So we say they're conjugated. Yet another addition to our protein knowledge. So proteins, when they're made, sometimes and often are in an inactive form. So inactive means they're not doing their role. An active form means they're carrying out the specific role they're designed to do. So we produce some proteins in an inactive form, and then something needs to happen to that protein structurally to change it, to activate it, to turn it on, essentially. And it sort of makes sense if you think about it. We don't want our body to have, a lot of enzymes in particular break down things, catalyze reactions. And there's going to be times where we don't want those reactions to take place. So it's good that we sort of have this key, this way of turning proteins on and off sometimes. Typically what happens, I suppose it can differ for different proteins, but this example with insulin down the bottom is a good example and a pretty straightforward one. So when insulin as a protein is first produced by the body, so insulin, by the way, is a hormone that helps your cells take up glucose. When insulin is produced, it looks like this, which is a big long chain folded into a specific 3D structure that's held together by 1, 2, 3 disulfide bonds. If we want to activate insulin, we have another enzyme that comes along, we call it an activating enzyme, comes along to this inactive protein, and it sort of makes two cuts, if you like. One here and one here. And it gets rid of a section of that protein so that it looks like the one over here. So in the active form of the insulin, you've got two separate chains now, whereas before we had just one continuous chain. So you've got two separate chains, and those chains are held together by 3 disulfide bonds. And you can see it clearly looks different in the active form to the inactive, and that difference in its structure has now made it active. This is just a bit of a definition type slide. So you guys might have heard of the Human Genome. The Human Genome Project was a project that went about sequencing the entire human genome, which is all of the genes within a particular cell or organism. A proteome is the protein version of that. So we call an organism's proteome the complete array of proteins produced by a single cell or an organism in a particular environment. You guys would have touched on protein packaging and transport a little bit when we did transport across membranes. So this is a nice detailed version of exocytosis, which is the process we use to take proteins that are made inside one cell that then leave that cell to travel somewhere else in the body. So what you need to be able to do is you need to be able to track and explain the pathway that a protein takes from the time it's manufactured within one cell and exits the cell to go and travel elsewhere in the body. And this is how it goes pretty much. So as you would know, in a cell you have a range of different organelles. One of those organelles is called your endoplasmic reticulum, and specifically your rough endoplasmic reticulum is covered in ribosomes. And we would also know that a ribosome is the place where proteins are manufactured. So step 1, polypeptide chain grows from a bound ribosome. Step 2, that polypeptide chain passes through a pore in the ribosome to enter the internal space. Travels through a pore in the endoplasmic reticulum to enter to the internal space of the endoplasmic reticulum. Whilst the polypeptide chain is in the endoplasmic reticulum, that's where it folds up into a 3D shape. When it's formed its 3D shape, it's loaded into a transport vesicle, which then exits the endoplasmic reticulum and travels to the Golgi apparatus, or the Golgi complex, whatever you might call it. The Golgi apparatus is an organelle in its own right, and its role is to receive those proteins from the endoplasmic reticulum, modify them in some ways, store them, and then eventually package them into a secretory vesicle, which gets released from the Golgi, travels to the plasma membrane of that cell, fuses with the plasma membrane, and then releases the proteins inside it via exocytosis.